So welcome to this month's ECSA webinars file. We are delighted to have Stephania here with us today. Stephania was the winner of the Peter Jones Memorial Award for 2021. And I believe the work that was in that paper is what we're going to see today. So Stephania is originally from Greece and has a BSc in biology, and then moved to the UK to study an MRes, and then started a PhD at Queen Mary's University of London in microbial ecology. She's currently in her final year, so dealing with the pressures of writing up, I imagine. But we're looking forward to seeing your work today. So Stephania, over to you.
Thank you very much. Let me start sharing. Now, unfortunately, my internet is a bit weird, so I will turn off my camera for the presentation. If I can find out how to do it. Stop. Yeah, there we go. So yes, so as make it smaller. As Jonathan mentioned, I'm Stephania Tsola, and I'm a final year PhD student at Queen Mary University of London. In 2021, I won the Peter Jones Memorial Award, so I thought it would be a good idea to sort of present the research I did there for the Medway Estuary for you today. Later on in the presentation, I will present some results from another member of my team and some ongoing work that they're still doing on the Medway Estuary. And also I'll talk to you about two of the major other projects that I'm working on as a PhD student at Queen Mary. So the title of my presentation is Methane Production by Dimethyl Sulfide Degradation along the anoxic sediments of the Medway Estuary in the UK. But before I go into more details about my project, I would like to have a look at these photos. What do you think they have in common? Well, surprisingly, the common link is actually dimethyl sulfide, or DMS as it's most commonly referred to. Seals and foraging seabirds are able to detect DMS, leading them to all the best feeding grounds. DMS also gives the characteristic smell to cooked seafood and the wonderful smell of boiled vegetables like cabbage and Brussels sprouts. It's also released during cheese and beer making, in the beer making processes, as well as paper production. So DMS is pretty much everywhere. But why is it important? Why am I studying it? Well, DMS is the largest source of sulfur in the atmosphere. So about 80% of the atmospheric sulfur actually derives from DMS. DMS also takes part in both the sulfur and carbon biogeochemical cycles and is considered a climate cooling gas. To help you understand this a bit better, I made this map showing the biogeochemical cycle of DMS in the atmosphere, water column, and sediments. DMS is considered a climate cooling gas because when it reaches the atmosphere, it makes aerosols, which can lead to cloud formation. So more clouds means less radiation reaching the Earth. This is known as the Clough hypothesis, which is it's generally a hot topic between DMS people because not everyone believes that DMS contributes to the atmospheric aerosols as much as some people believe. Nevertheless, though, it's something that does happen naturally in the atmosphere. Now looking at DMS production, overall it has many production pathways, especially in sediments, like methane methylation and sulfur-containing amino acids. But its primary precursor is dimethyl sulfonneopropionate, or DMSP. DMSP is produced by plants, like the salt marsh cordgrass spartina, that's how it ends up in the sediments, but it can also be produced by phytoplankton. This phytoplankton can then be cleaved by this DMSP is then cleaved by abiotic processes or bacteria to DMS. And this is what happens in the water columns. So phytoplankton releases DMSP when it's being grazed by zooplankton. That then is cleaved to DMS, which is then detected by seals and foraging seabirds, hence my previous slide about the leading them to all the best foraging grounds. Looking at the DMS product, the thing I would like to make known here, and I would like to emphasize, is that despite DMS being about 80% of the total atmospheric sulfur, only about 10% of the produced DMS is actually released in the atmosphere. So the other 90% remains in the water column and sediments, where it's degraded either aerobically or anaerobically. Generally, its aerobic degradation is better studied, potentially because it's easier to set up aerobic incubations, but I'm not really sure about that. But its anaerobic degradation, so this part of the graph, is actually my main focus. So this is where I'll be focusing on for the rest of my talk. So the DMS degradation in anoxic sediments. Most studies on this topic were done 30, even 40 years ago now, and were primarily culture-dependent studies. So inhibition experiments and isolation studies where no Illumina sequencing was done later on. However, that research was able to teach us that methanogenic archaea, like methanomethylloborans in the top photo, and sulfate-reducing bacteria, or SRB, like Desulfosarcina and Desulfococcus in the bottom photo, are able to degrade DMS. Now, these organisms degrade DMS and produce two important greenhouse gases. Sulfate-reducing bacteria, or SRB, produce carbon dioxide when sulfate is available in the environment, and methanogens produce methane, a greenhouse gas with 28 times the global warming potential of carbon dioxide over 100 years. Now, even more interesting is that methane is actually often presented in media as a key climate change player. So it's surprising that not much is known about this methane production pathway. So knowing that the anaerobic degradation of DMS is not well studied and that it leads to the production of two greenhouse gases, we set out to explore the microbial DMS degradation in anoxic sediments. More specifically, we wanted to investigate the potential for methane production that these sediments have, as well as the diversity of DMS degrading microorganisms. Now, you're all probably wondering, okay, we understand what they're trying to do, but why did they choose to do this in an estuary instead of collecting sediments from literally any other environment? Well, estuaries are model ecosystems for studies like ours. They are partially enclosed bodies of coastal water influenced by the tides, where low salinity water from rivers and streams mixes with high salinity water from the oceans. Estuaries generally cover a small area on Earth, but both DMS and methane have been detected here. This, coupled with the salinity, hence sulfate gradient they are characterized by, works in our favor when trying to answer questions like ours. So, in the first year of my PhD, in November 2018, on a very, very cold British rainy day, we set out from East London to collect sediment using cores like these. And we went to the Medway Estuary, which is in the southeast of England. You can actually see it in the small map at the top. There we sampled 3 locations along the sulfate gradient of the estuary. You can see it. You can see them numbered in the map. Number 1 was the freshwater site from Maidstone. Number 2 was the brackish site from Medway Bridge Marina. And number 3, we had Grain, which was the marine site, and you can actually see it's at the mouth of the estuary. When we came back to the lab, I set up incubations like these. I added artificial seawater, the salinity of which was matched with each individual sampling site, and sediment to serum bottles. Each bottle was made anoxic with oxygen-free nitrogen, and the samples were amended with DMS. Now, DMS amended was done incrementally to try and avoid any potential toxicity to the microbes. So I would add DMS, wait for it to be degraded, and then add more, so on and so forth until the end of the incubations. So I also had two types of controls. The first type of control contained artificial seawater and sediment, but no DMS. This was done to try and monitor for any potential methane production not derived from DMS. The second type of controls contained artificial seawater, DMS, and autoclaved sediment. This helped us calculate how much DMS can be lost because it forms complexes with the sediments. The bottles were then incubated at room temperature, and gas chromatography was used to monitor DMS degradation as well as methane and carbon dioxide production. The incubation experiments were then terminated when methane production reached stationary phase. The bottles were opened and the water phase was collected for dissolved inorganic carbon and sulfate measurements, whereas the sediment was used for microbial diversity analysis. More specifically, I amplified and sequenced for the 16S rRNA gene that corresponds to all bacteria and archaea, for the MCRA gene for all methanogens, so methanogenic archaea, and the DSRB gene for all sulfate-reducing bacteria. So let's move on to the results now. Here you have the averages for DMS degradation with black on the primary axis and methane production with orange on the secondary axis. Every DMS spike is when I added DMS to the incubations. Out of the 3 sampling sites, excuse me, the marine incubations lasted the longest at about 64 days, and the brackish lasted the shortest time at 22 days. Now, DMS degradation to methane follows this chemical formula. So 2 molecules of DMS get converted to 3 molecules of 2 molecules of DMS get converted to 3 molecules of methane. And knowing this, I was able to calculate the DMS degradation efficiency, which was about 40% in all sampling sites, regardless of sulfate availability. But as I mentioned earlier, SRB also degrade DMS. So let's look at how they do it. Sulfate-reducing bacteria degrade 2 molecules of DMS to 4 molecules of carbon dioxide. So this could explain, if they also degrade DMS, this would explain the low efficiency that we're seeing at 40%. So let's look at the carbon dioxide results, which have just popped up. Here you see the total amount of carbon dioxide produced at the end of the incubations. And it's about, for the freshwater side, is the highest at about 5 micromoles per gram of sediment, which is extremely low compared to the methane, which is about 40 micromole per gram of sediment. So what's happening here? Are the sulfate-reducing bacteria actually degrading DMS? And if they are, what's happening to the carbon dioxide that should be produced? So in order to try and answer these questions, we looked at the amplicon results, the amplicon sequencing results. So here you have the MCRA results, so for all methanogens. Graph A shows the relative abundance of the methanogens at genus level, whereas graph B takes the same amplicon data but presents it on a PCA, or principal component analysis plot. Overall, we see methanogens increasing in the sediment treated with DMS when compared to the untreated original sediments. That's what ORI stands for. This is seen in the PCA as well, where we see the sampling locations in different colors. So red for marine, blue for brackish, and black for freshwater sediments, going from loosely clustered to completely separate. In the little table you see in the corner, that you can see the Spearman's rank correlation analysis I did, which helps us understand that changes along PC1, so the x-axis, are due to DMS and methane changes, whereas PC2, so the y-axis, the changes we can see are actually related to sulfate before and after the DMS incubations. Looking at the relative abundance graphs now a bit more closely, we actually see that in the freshwater sediments, a known freshwater DMS degrader, methanol-methylovorans, increases to 66% after the addition of DMS. In the brackish sediments, another known DMS degrader, methanolobus, can be seen increasing to about 73%. Lastly, in the marine sediments, we see methanococcoides completely dominating after the addition of DMS. Now, this result is interesting because methanococcoides has not actually been found to degrade DMS before, and we can actually see it increasing in all three of the sampling sites with dark blue. So this is it's interesting, and it's something that we're still trying to find out more about. Now, moving on to the sulfate-reducing bacteria data. Here, making conclusions isn't the easiest because only about 240 SRB exist in pure culture. So assigning taxonomy isn't always the easiest. Regardless, though, we can see some changes between DMS incubated sediments and the originals. Different delta protobacteria seem to be increasing in the freshwater and brackish sites after the addition of DMS. And in the marine sites, we see an uncultured lineage increasing after the addition of DMS. A similar pattern of separation can also be seen in the PCA graph on the right, with most changes correlating to sulfate, which I guess makes sense since we're talking about sulfate-reducing bacteria. So overall, SRB appear active in the sediments and degrading DMS. But if that's indeed the case, where is the carbon dioxide that should be produced? And if DMS degradation by SRB is sulfate dependent, as the name and the PCA graph suggest, how come SRB appear active in the freshwater sediments as well, when sulfate there is practically non-existent? To answer both those questions, here are the bacterial sequencing results. Here it's for the 16S, but I've removed the archaea, so it's just the bacteria. The changes in relative abundance are definitely not as clear-cut. Even in the PCA plot, no clear groupings can be seen after the incubations, besides for the freshwater sediment with black, where you can see some minor clustering rather than a spread of samples as it is for the other incubations. Also, these changes seem to correlate with all parameters we checked against, so no conclusion can really be reached. But the major change we did see, though, was for this genus, Sulfuromonas. Appearing with a star. Sulfuromonas is a sulfur oxidizing bacterium known to autotrophically fix carbon dioxide. And in our incubations, we actually see it increasing after the addition of DMS, suggesting it is the reason why so little carbon dioxide was measured. Sulfuromonas has also been shown to be a member of a cryptic sulfur cycle, where hydrogen sulfate, in this case continuously produced by DMS degradation, is recycled to sulfide and sulfate, making it available for the sulfate-reducing bacteria. That means that this could be something that we see in the low sulfate environments like the freshwater sediments. So, in conclusion, this project helped us show that DMS is indeed a methane source in estuarine sediments, and that there's a sulfate-driven niche partitioning of methanogens in the estuary, where we have methanomethylborans taking over in the freshwater sediments, methanolobus in the brackish, and methanococcoides in the marine, which is not actually reflected in the methane yield, which is about 40%, regardless of sulfate availability. Interestingly, methanococcoides was also found throughout the sulfate gradient, even in the freshwater sites where it hasn't actually been found before, since it's a saline methanogen. So this warrants future research. Lastly, DMS was also found to be degraded by SRB, and we were able to detect the potential existence of a cryptic sulfur cycle in freshwater sediments. This suggests SRB activity even in low sulfate sediments, like our freshwater ones. Next, I'd like to present some ongoing and future work myself and another member of my lab team are currently doing and will be doing in the future. So the wonderful Susie seen in the photo is continuing the Medway Estuary experiments currently, and it seems to be having some very cool results. She's currently running inhibition experiments, inhibiting methanogenic archaea in some incubations, and sulfate-reducing bacteria in others, along the sulfate gradient of the estuary, as I explained earlier. And this will help us fully understand methanogen and SRB interactions. Then the plan is to do some salinity and temperature experiments as a climate change model to help elucidate if higher salinity and temperature in, for example, the freshwater side would or could cause a shift in the DMS degrading populations. Lastly, she's planning on completing some stable isotope probing incubations, so incubations containing carbon-heavy DMS. This will help us find the active DMS degraders in the estuary, and using the RNA from the SIP experiments, we could gain valuable insights into the metabolism of the DMS degraders. So with that, I come to the end of my Medway Estuary story. But as I mentioned in the beginning, I'm still doing a PhD project. So I'll be briefly talking about some of my other projects now. First, we have the Baltic Sea project. So the Baltic Sea is the largest body of brackish water in the world. So it's ideal to monitor SRB and methanogen populations, kind of like an estuary is. Secondly, though, massive phytoplankton blooms can be seen here, and they can actually be seen by satellite as well, and they appear every spring and summer. So these large blooms actually means huge amounts of DMSP are released in the water columns, which is, as I mentioned earlier, a major DMS precursor. So, knowing this, I sampled 3 locations in the Himmerfjärden estuary. I'm probably saying that completely wrong, so I apologize. Starting near Stockholm at the very top. I don't know if anything is like you can actually see the sampling sites, but I started up in Stockholm, which is near a water treatment plant with H5, and then I started sampling down the estuary towards the open Baltic Sea. I also sampled 7 depths in all locations, and you can actually see the salinity of which in the tables, and this is what I use to set up my incubations. Preliminary results from here are showing higher DMS degradation in the sites with higher anthropogenic and phytoplankton activity. The two are related since higher anthropogenic activity means higher nutrient levels are released in the water columns. So I guess it makes sense that higher anthropogenic activity means higher phytoplankton activity, so higher DMSP levels. But it's still very interesting to see it affecting the DMS degradation in the sediments as much as it is. So I'm still looking into that. I've also done some metagenomics for certain samples. From the metagenomics, I actually got 4 very high quality metagenome assembled genomes, or MAGs. All of them were from Methanolobus, which you probably remember from the Medway Estuary brackish sites, where they dominated after the addition of DMS. So I made this, I very quickly made this heat map, which shows the completeness of various methanogenesis pathways. So I looked through all the methanogenesis pathways and certain other genes that take part in methanogenesis, like, for example, the MCRA and the MCR regeneration complexes. So blue here means high completeness, so all genes from the pathways are present, whereas orange means more of, none of the genes are present. Interestingly, none of the MAGs have genes associated with DMS-dependent methanogenesis. So this could mean that the genes don't actually exist in the databases, so they could potentially be novel, or that DMS-dependent methanogenesis uses genes from other methanogenesis pathways, like for example, the trimethylamine methanogenesis at the very top, which we can actually see be complete in all 4 of the MAGs. But I'm still looking into this, and to understand exactly what's going on and to find out more about the metabolism of DMS degradation, I'm currently analyzing metatranscriptomics samples. They're the same samples that I sent for metagenomics, so it'll be interesting to see if what the copy numbers show and if I can find any DMS-dependent methanogenesis genes there. So that's still ongoing. My second large experiment is on rivers. Rivers are freshwater ecosystems, of course, and considered methane hotspots. And also large amounts of methane are actually released from rivers every year. And weirdly enough, some papers are actually suggesting that a warming climate could mean more methane being released. So specifically, there's a 2015 paper that demonstrated that a 2-degree rise could increase the methane emitted by rivers by 8%. So I wanted to understand if DMS degrading microorganisms exist in rivers, which they must, and what proportion of this methane could be derived from them. So I ended up sampling 4 sites, 2 rivers, Pant and Rib, which were characterized as gravel-dominated riverbeds, and another 2 rivers, Nadder and Medway, which are sand-dominated riverbeds. Now we understand what the riverbed looks like by measuring the grain size of the riverbed. So a bigger grain size means that they are gravel dominated, whereas a smaller grain size means they're sand dominated. So here are the sizes. I had forgotten I put them there. So here are the sizes for the grain size between sand and gravel. So what we do find is that sand-dominated riverbeds have a methane yield higher than 90%, which is huge, especially compared to the 40 to 50% yield we see in the gravel-dominated riverbeds. So this is probably a result of permeability. Sand is less permeable than gravel, meaning less oxygen travels down the sediment. So this benefits the methanogenic archaea because they are obligate anaerobes, so less oxygen means they can function more and better, and they can have a higher abundance. Despite the differences in yields, though, methanomethylborans, as you probably remember from the Medway Estuary experiments, a known DMS degrader, can be seen increasing in all rivers and are actually completely dominating after the DMS addition. Interestingly, though, we do actually find methanococcoides in 2 of the rivers. And since methanococcoides is primarily found in saline environments, I'm still looking into what is happening here. Lastly, I'm currently analyzing my data for sulfate-reducing bacteria. And they might shed some light on the cryptic sulfur cycle idea that we had for the Medway estuary data. So that'll be interesting to see. I'm doing that currently. So with that, I think I'm going to finish here. Hopefully I didn't confuse you too much with all the different experiments. And in closing, I'd like to thank everyone that has helped me along the way, especially my supervisor, Oscar, Queen Mary University of London for awarding me a studentship to complete my PhD with them, and the Estuarine and Coastal Sciences Association for the Peter Jones Memorial Award and the opportunity to present my work with you today. Thank you all for listening.
Great. Well, thank you, Stephania. If there are, we've got plenty of time for questions. So if there are any questions, can I encourage you to write them in the chat, and then we can go through them. In the meantime, whilst people are writing questions, I have one, if that's okay.
Yeah, of course.
So I was wondering, has anyone looked at DMS in restored wetland environments? Because obviously you could potentially have benefits through restoration, but also with the transition from what used to be an agricultural field to an intertidal environment, that also might potentially have some negative effects. I was wondering if you've got any comments on that.
That's a very good question. The answer is no. They've primarily looked at DMSP, so the precursor of DMS, but because it's such an interesting question, we actually have a postdoc that's just started in the lab, my supervisor does, that will be looking at peatlands and wetlands to try and see what goes on with DMS in those ecosystems. And they'll be doing some SIP experiments as well. And I think Susie, after she's done with the Medway Estuary stuff, she'll be doing some peatlands at least. So so that'll be interesting to find out if what's going on there.
Oh yeah, definitely. It'd be good to see what results come of that. So we're still waiting for a couple of questions to come through.
I'll stop sharing so I can see the chat.
Everyone's very quiet.
That's okay. It was a lot of information, I think.
Do you have any thoughts on how obviously there's a big drive at the minute for making people aware of things like blue carbon. What can we do to spread the word about DMS?
I mean, honestly, it's a question that I often ask myself, because it's everywhere, and it's the smell of the sea, to be quite honest with you. Like the characteristic smell that you always have at the seaside, it's normally DMS that you can smell, but it's not. It'll be nice if more people got in researching it. I think that's the solution. If more people were researching it a bit more, I think it would help people understand DMS more. And also, it's funny because, like, in America, DMS is used as a food additive. So yeah, it gives like an umami taste, and but still no one really knows about it. So yeah.
I like the idea of the smell of the sea. So there's been a couple of, can you see the chat?
Yes, I can.
Okay, so we've got a couple of comments. So first of all, yeah, the video will be on our YouTube channel later today, for anyone that wants to watch.
Yeah, and if you have any questions, then I'm more than willing to answer, either on Twitter or my email account.
Okay, so there's been a couple of comments. I don't know if you want to come back on these.
Yeah, so wonder if archaea are disrupting other biomes' pathways. That's really interesting, and that's actually part of Susie's experiments now with the inhibition experiments, where basically we're inhibiting, well, she's inhibiting the methanogenic archaea in some incubations to check if this changes anything in the environment. But these archaea are naturally found in the sediments, since DMS is there. So I don't think they will be disrupting the biomes, since they're naturally occurring there. And you might be interested in the mass program at Mayo Clinic. Archaea. Oh, I'll look into that. I'm not aware of the program you're referring to. I looked into that. Thank you.
Okay, well, if there's no other further comments, then perhaps we'll leave it there. So thank you very much, Stephania. Before we go, I just want to make everyone aware of our next webinar, which will actually be again in March. We have moved April's webinar to March because the date clashes with the ECSA AGM. That's a lot of letters there, but ECSA's AGM, so we're gonna have to move it a week earlier. But it will be given by our very own Martin Wilkinson, who is the ECSA treasurer, and Martin will be talking about what 250 years of studying seaweeds in the Firth of Forth can tell us. So if seaweed's your thing, we look forward to seeing you there. If not, thank you for everyone who's attended today. Thank you again, Stephania.
Thank you.
And I look forward to seeing everyone soon. Thanks, everyone.